Hi, so I wanted to clear out some doubts that you may have about haploid and diploid cells and at what point the diploid original germ cell becomes haploid in meiosis. So this is a very tricky question. And the reason is because the definition of chromosome is very tricky. Okay, so when we're looking at this chromosome, we know its homologous pair is this chromosome. So this chromosome can be chromosome 3 that came from dad, and this chromosome is chromosome 3 that came from mom. Now, in interphase, when these chromosomes duplicate, they attach to their clone, okay, right over here. But they're still, even though now they're two copies of that chromosome, they're still called just one chromosome. And the reason why it's still just one chromosome made out of two chromatids is because they're copies. You haven't created any new piece of information. All you did was you duplicated, right? And that's when a lot of students get confused. This sister chromatid, this X form, is made out of two chromatids of the same chromosome. Okay, and that's why it's called a duplicated chromosome. We don't call it two chromosomes. We call it a duplicated chromosome. Okay, so at this point, you're still diploid. And the reason why you're diploid, D meaning two, is because in the same cell, you still have the chromosome from your mom and the chromosome from your dad for that pair. You still have the pairs of homologous chromosomes. Okay, you've got the set from mom and you've got the set from dad. That makes you diploid. You've got two sets, one given to you by mom, one given to you by dad. Now, what happens at the end of meiosis 1 is that those homologous chromosomes are separated. They're the ones that are separated. So in one side, in one of the daughter cells at the end of meiosis 1, you're going to get one of the chromosomes, maybe dad's. And in the other daughter cell of the other product of meiosis 1, you're going to get the other chromosome. So you've separated the homologous pair. That means you no longer have two sets, one given to you by mom and one given to you by dad. You have separated them and randomly assorted some homologous chromosomes to one side and the other pair to the other side. You've randomly assorted that to these new daughter cells. Now, even though you still contain the copies of the chromosomes, you still contain the chromatids, you are now you're now haploid because you do not contain the diploid set. Now, you can't stay here. Meiosis 1 is not suitable. It's not going to be a cell that survives. You can't stay here. You need to, by obligation, go into meiosis 2 and finish off meiosis 2 because you still have these sister chromatids that you need to divide, where one chromatid is going to go to one side and the other chromatid is going to go to the other side. But now all you're doing is you're dividing the chromatids. So you're kind of like distributing the clones. You're distributing one clone to one side and the other clone to the other side, but you're still giving the same information, the same relative information, like one chromatid to one side, the other chromatid to the other side in each one of these daughter cells. So it's a little bit complicated, and that's why we don't actually go into detail on this, because you definitely would in a little bit of an advanced biology class. But the idea to always, always, always remember is that as long as you've got the two sets of the homologous pair, you're diploid. The minute that you no longer have that set of the homologous pair, you are now haploid. So the minute you separated the homologous pairs of chromosomes, you are now haploid, and the only thing you need to do now is distribute your chromatids, okay, into each one of your daughter cells, producing 4 haploid cells. So here you would have 2 duplicated haploid cells. So this would be a haploid cell that has twice as much information as it needs. This would be a haploid cell that has twice as much information as it needs. It is not a diploid because it doesn't have the 2 homologous pairs of chromosomes. but it still has twice the information it needs, so it'll be dividing once more so that it can now have 4 haploid cells each with half the amount of chromosomes. Okay, now if you want to think about this in the sense of oogenesis, I was also asked about that. Okay, a lot of people get confused at this point where you have the secondary oocyte, which is right in the middle of metaphase 2. So you kind of stay kind of like right in the middle over of here. You're not you're not finishing meiosis 2 just yet. Okay, and you're only going to finish meiosis 2 if, in fact, your cells, your secondary oocyte cells, come into contact with the sperm. So the minute that the sperm actually just touches and kind of breaks up the membrane of this cell, the cell will immediately go into meiosis 2, will immediately finish meiosis 2, let go or eject the second polar body. So it'll eject one of the cells that are not going to be used, they're just going to be disregarded. And when they do that, when they eject that cell, then the nucleus with all of the DNA of the father can enter this full ovum that has just recently become an ovum because it's ejected the divided cell that resulted from the end of meiosis 2, from the product of the end of meiosis 2. And now the sperm cell can, the nucleus of the sperm cell with its DNA can fully enter this cell and merge with the DNA of the egg, creating a diploid cell. So it doesn't happen simultaneously. The sperm reaches the egg, and the minute that it reaches the egg gets the signal to finish meiosis 2. It lets go of the second polar body that is not necessary. Now it has the haploid set of chromosomes, and then it can merge with the haploid set of chromosomes from the sperm to create a fully fertilized egg and a diploid zygote that will go on to become a baby. So hopefully this makes sense.